My pleasure. Yeah, thank you all. It's been a wonderful 2 weeks, fantastic experience, and it's been wonderful seeing all the work that you've all been doing, and it's been an honor to be a part of this. So I'm up here representing a large group of people here that worked to do the canopy work. So I'm going to be presenting some of the stuff that Wendy and I focused on collecting leaf samples from the trees and other samples for other taxa as well as the work that Jen and Steve were focused on collecting epiphytes in the canopy. Before we even got here, a tremendous amount of work was done by the USM team led by Ramad and Abu, putting in these forest plots. A lot of us have been dependent upon those plots for the tree identification. That was a real important foundation for our work, as well as identification of these species. We're not tropical forest at Penang Hill tree experts, and so their contribution to this was just invaluable. So this is the first canopy survey of Penang Hill, and possibly Malaysia, as far as I'm aware of. So this is a real important first step, I think, in a long-term effort, I hope, to continue this type of work in the canopy. As you all know, most of the foliage and the trees and a lot of the life in the canopy is really driving what's going on in these ecosystems. Most of the photosynthesis, a lot of the biodiversity is actually found high up. And so in order to really understand the biodiversity of this place and the ecosystem ecology and everything, we really need to get up into the canopy. And so it's been an honor to contribute to that. And here's Wendy collecting some leaf samples. As you can imagine, climbing these trees can be dangerous, and safety is our primary concern as we're climbing up into these trees. Not only making sure that the trees are structurally sound, but protection from snakes or wasps or insects, other insects that might bite you, or poisonous plants, as Wendy and Bryson found out. So Tim Kovar here, who had to leave this morning or yesterday, and Kay, we're really critical in ensuring our safety and protecting us. And a huge thanks to Lala and Zul. They accompanied us every day and really made a huge difference in being able to allow us to do our work effectively. So our team, which consisted of myself and Wendy, Steve and Jen, Bryson and Kevin, we did a total of 58 climbs over the course of these 2 weeks. A total height, we estimate about 1,596 meters we climbed total. Here I am in Shorea bracteolata tree on the habitat. It's the platform tree as you approach the shop. It's listed as an IUCN endangered species. It's Meranti Paang is the Malaysian name for it. It's a rare species. I think there's only one that we're aware of in the habitat. It was a real pleasure to climb this tree. To put this the amount of meters climbed in perspective, we have you can see on the left there the stacked trees of 1,596 meters compared to the Petronas Towers in KL and Penang Hill at 451 and 833 meters. So it was quite an enormous amount of work. I wish we could have climbed more trees, but that will require more time and visits. So Bryson and Kevin led up the mammal surveys, canopy trapping in the canopy. You can see Bryson showing off there with Kevin. And you already heard from Kevin about that. Jen led up the epiphyte survey work in these trees. There she is climbing a tree, collecting whatever epiphytes we could find. Some of the trees didn't have a whole lot of epiphytes. There was most of the trees had some epiphytes, but some of them were absolutely loaded with epiphytes. This one, which is right off of the canopy walkway, which was just an incredible tree, loaded. Here's Steve doing a survey and collections in that tree. It was quite nice being able to actually climb straight off of the canopy walkway rather than having to climb all the way from the ground and save us that effort. I don't think Steve was able to survey every single part of this crown. That would have probably taken 2 weeks in and of itself. So there's more work to be done. And I'm sure there's a lot more species that could be collected. So Wendy and I were focused on the leaf samples, trying to collect samples from the top of the tree where the sunlit foliage is at. And in cases where there was a difference in the morphology between the top and the bottom of the crown, we would also collect the lower foliage as well. Sun versus shade foliage, morphological differences, which has real important effects on the physiology and functioning of the tree as well. Here's Abu and Ramad helping with the plant identification of the trees and vines that we collected. Their help was invaluable, it was critical. Also we had some help from Rexy with the orchid. There on the right. It was critical for the orchid identification and help with Jen. And we had a lot of help with plant pressing for the herbarium specimens. Here's Meg showing some of the Jason students how to do the plant pressing, and so we really appreciated the help of them, and Wendy's mom, and Nancy, and several other folks that helped with the plant pressing. We also collected some samples for Meg to use with the students for herbivory, and she's found differences between the tops and the bottom of the canopy in other studies, and that was something that she was able to do this time as well. And I'm looking forward to hearing the results of what she found. And while we were up there, we wanted to take advantage of our being up in the canopy. We collected moss and soil samples and other substrates that were up in the canopy for tardigrades, and here's Randy looking at some. I don't think he's had a chance to actually analyze any of those samples that we collected, but we may be in for some new discoveries there as well. And we also collected a subset of these leaves and other samples for Dina and her crew to look at the algae and microbes, and so that's going to be a tremendous amount of work, I'm sure, going forward to analyze all those samples as well. And we helped Durrell with his mosquito crew in setting up some traps for the mosquitoes. Here he is. That's not the CO2, is it, Durrell, or is that the white? Light. Oh, UV, yeah. So in all, the USM crew found 53 different tree species in the plots, representing 27 families. We climbed 13 different species, representing 10 families. We wanted to maximize the diversity. Obviously that's a small subset, but because of the enormous amount of time it takes to access these trees and climb them, we couldn't climb all of them. We also, Jen and Steve collected more than 50 samples of epiphytes and lianas. They are still in the process of identifying a lot of those, but so far they've identified 15 orchids, 8 ferns, and 4 different lianas or climbing vines. Here's an example in one of the trees, this Dacrydium elatum, a podocarp tree, where you see the difference in the foliage from the top to the bottom of the canopy. You can see. And this is really important because a lot of herbarium specimens are collected from lower down, and if you don't understand the difference in the morphology, you could actually see misrepresent or misunderstand what species you're looking at, and it's really important to characterize the full gradient and morphology for these specimens. And Natalie already talked about like the DNA analyses of these herbarium specimens, but herbarium specimens are also really important for to be used as kind of time capsules. They represent a snapshot in time of not only the presence and the distribution of these species, but also they contain really valuable information. The leaf chemistry, the leaf anatomy and morphology is really important so that you can track things over time, and you can go back 100 years or more and look at herbarium specimens, look at the stable carbon isotope composition, for example, and the stomatal density, to look at how plants have changed in their response to climate change, for example, and how environmental change is affecting them. So these are really critical specimens that can be used going into the future and documenting not only where species live, but also how they are functioning. So this is just we just scratched the surface of these samples here. Dr. Mansour at USM did a review of the Penang National Park flora in 2016 and found a total of 521 plant species, 110 families, and 334 genera. 250 of those were trees. So there's a lot more work to be done in collecting these herbarium specimens. We're just started, and hopefully we can continue this into the future. And thank you to the cockerels and Habitat and everybody. It's been amazing. Thank you so much.
Thank you, Anthony. Gosh, a great example of so much collaboration.